give a lecture on using bioinformatics to extract information about various aspects of proteins that you may be interested in working on. You know, I think as a result of this huge sequencing effort that took place back in the 90s, we've gained so much information that we don't really know what to do with. And so out of that, bioinformatics sort of began to rise exponentially, where people have generated complex computer programs to allow them to go in and mine the genomes of various organisms. And Dr. Babbitt really is one of the best in the world at doing that. I've had the pleasure of working with her and her colleague, Gemma Holliday, on trying to annotate this huge superfamily of metalloenzymes that many of you may be aware of, called the radical SAM superfamily. And they discovered that there are well over 48,000 individual sequences of reactions, of proteins that are catalyzing myriad reactions, most of which we have no idea what they are. But based on what we know so far, a lot of them are incredibly exciting. So they are giving us plenty of material to work on. 48,000, we have a lot of work to do. So, in her talk today, and then in the workshop later on today, and then in Bioinformatics 2 and Bioinformatics 3, they will start to teach us how to go in and use some of the resources that are out there for mining the various genomes of many organisms. And some of these resources are resources that they develop themselves in their own laboratories. So thank you very much. Patsy, welcome.
Thank you very much for a very kind introduction. Can you all hear me okay back there? I love the weather. It's absolutely gorgeous, and I have a lot of hay fever today, so I'm going to try not to start coughing in the middle of this, but we'll see how I do. I'm very pleased to be here. I'm going to talk about an introduction to bioinformatics, if I can get this to work.
Jared broke it.
It's a green light on here. Ah, there we are. I just had to find it.
It's calibration.
Thank you. So bioinformatics is actually many, many different fields. And I can't possibly begin to cover them today. I could make you a long list, but that would take up 90 minutes. So I'm going to focus on some of the fundamental principles for inferring molecular function, because that's what Squire told me would be perhaps of special interest to the group here today. I'm also going to really talk about very fundamental principles behind the major tools, because I think then that arms you with a little more information to use the tools, which are actually very easy to get at, and they typically have very good tutorials. That means that many of you will be much more sophisticated than much of what I'm going to talk about today, and if you'd like to go to sleep or play ping pong or something, that's okay too. I tried to hit it as well as I could to be quite basic, but also give some a little bit more in-depth information. So the function, prediction, and annotation problem. What we really want to understand is how proteins evolve to do the jobs they do, in particular enzymes. And my group works principally on enzymes because they do chemistry. And that means we can get quite explicit mappings between structural features, either seen in sequence or structure, and functional features. And that's really important for doing that kind of structure-function mapping. And a lot of the way we think about it outside of bioinformatics, but when we're really doing our science. So one of the problems is that we have now more than 50 million sequences in the major public databases. This is still increasing at a logarithmic rate. It was 29,000 a year ago. 29 million a year ago. There are also billions of metagenomic sequence reads for community organisms, whether they're bacterial organisms in specialized environments like acid mines, or whether they're environments living in the human body. This is also growing by leaps and bounds. We can't culture many of those bacteria, and so we're going to have to do a lot of our functional inference in the data mining and trying to even figure out the targets that we want to look at experimentally using bioinformatics approaches. So I agree that this is timely and important for education in general in the life sciences and chemistry and physics. So the proportion for which annotations or known functions can be experimentally annotated is becoming almost vanishingly small. We simply cannot do enough experiments to keep up with the data. And that really means that we do have to use more automated approaches to do that. The problem is our best information comes from experiment. And so how do we leverage that information to tell us the most about what all these proteins are doing? And then there's a huge issue of the open reading frames that in terms of misannotation. And that's just a scary and alarming issue. We published a paper on it a few years ago that tells us that for some protein families, misannotation levels are over 90% in the major public databases. And that's not for everything. It's for the non-trivial problems, but it's still an important issue. Today's main topic, which I'm framing everything around, is what we call homology-based analysis. Homology meaning referring to evolution of proteins from a common ancestor. And that theme is really an important underlying assumption about what I'm going to talk about. The assumption says basically that sequence conservation infers structure conservation, which infers functional conservation. And that's the basic paradigm that we use when we run a blast, when we compare structures, when we map protein data to other kinds of data like expression data or genome context, when we're trying to make inferences knowing something about protein A to tell us something about unknown protein B. It's a really important observation. I think it holds in very well in most cases. As we get to very divergent proteins, the non-trivial cases of function prediction, those are the unknowns that we don't know the answers for, and they're the hard ones typically. It gets very, very complicated, and the details really make this very difficult. The assumption is, of course, what's conserved over evolutionary timescales is going to be functionally important. And the application is really in comparison of sequence and structures to identify homologs and then asking questions about the nature of the knowns relative to the unknowns and trying to tease out something important. I don't have to get through all these slides today. I put a number of points in my slides that are posted now that you could just follow. I've got some resources and papers you can look at. So if you have questions, it's fine to stop me. I may try to keep some of it going just so I can get through a fair bit, but I'm happy to have this to be interactive. I need to give you a very, very short primer on molecular evolution and give you a couple of important definitions that you're going to see all the time. One is a homolog, of course, is those are proteins that share, or genes. I'm going to talk about proteins today, so I'm going to skip saying genes. I'm just going to say proteins. I do know we go through the genes to get the proteins. So they share a common ancestor by descent or recombination. Orthologs refer to similarity as a consequence of speciation. And what that means is that as species diverged, the proteins in those genomes also were carried through that speciation, and typically those proteins have similar or the same function. But there's nothing inferring function in these evolutionary models, so that it isn't the case that all orthologs have the same function. In fact, they can change quite a lot. All orthology speaks to is the mechanism of evolution. And it's just an important distinction. I'm not going to go into the details here. Likewise, paralogs refer to similarity between descendants of a duplicated gene. That is, you have an ancestral gene, it duplicates, and then since this function is still viable, it may evolve a different function. These two proteins are still going to be related. Typically, we can identify orthologs, and there are lots of good databases that do that, and distinguish them from paralogs when we're talking about fairly closely related species or fairly closely related proteins. When we get to very divergent proteins, where sometimes we don't have a lot of good linkers in between two very, very divergent proteins, it's much, much harder to tell what's going on. And you'll see why in just a second. Then, of course, we have convergent evolution that has a similar function, but not ancestral, not descendant from a common ancestor. So one of the absolutely fundamental aspects of the kinds of methods and approaches I'm going to talk about today is when we make an alignment, when we look at a blast output, we make a multiple alignment, we build a phylogenetic tree, we're making the assumption that all those proteins are related to each other evolutionarily. And sometimes it's easy to forget that if our primary way of thinking about, say, enzyme function is really at the functional side. So we're thinking about mechanism and saying these two proteins have the same mechanism, but if we don't step back and ask, are they evolutionarily related, they may come from different folds. So they may use a different structural strategy to present that mechanism. And I'm talking about the guys that have the same structural strategy, basically. And that's just a fundamental principle in this area. Some assumptions that are really important. As I said, phylogenetic reconstruction assumes homology. Both the mechanisms and rates of divergence are issues that we go after when we build phylogenetic trees. And you have to have a multiple alignment of homologous sequences to build a tree. And how good your alignment is determines how good your tree is. Now that's pretty trivial for the kinds of questions that these methods were initially developed for. We are talking about closely related species where you can align the genes or the proteins and there are no gaps that are forced in the alignment. Then you can actually build a very, very high confidence tree, and the mathematics are robust for that. The work that my lab does, and that I think increasingly many other people are doing for looking at unknowns, is we're looking at very, very diverse proteins. And sometimes we can't get good alignments. Our methods, how far we can see in sequence space are just not good enough. And so we have to use structural information to correct our alignments, for example. So that's really an important issue as we start looking at these very, very large data sets. Couple of other important points. Evolutionary history is accessed only using contemporary sequences. You know, sometimes we think, well, we have a sequence from archaea and a sequence from a prokaryote and yeast, and we kind of think about the prokaryote and at least yeast as sort of more recent. Well, they're all contemporary. And trying to infer what the sequences in these interior nodes look like can be really difficult. Again, people have done ancestral reconstruction to try to compute an ancestor. It works quite well when you're talking about closely related sequences. As you start to get more diverse, you're getting thousands and thousands of equally good, mathematically good solutions. And so that becomes, I would say, for the kind of work, some of which I'm going to show you today, we can try to build ancestral sequences, but I don't think they tell us a thing about evolution. They tell us something about properties that might be general to those contemporary proteins. It gets very difficult when you go a lot further. When we build phylogenetic trees, simplifying assumptions don't hold. All of our methods, all of the straightforward methods, there are certainly more sophisticated ones that don't have this problem, but the assumptions are that the changes at each site or in different copies of a gene are independent. We know that's not true. Do all the sites change at the same rate? We also know that's not true. Do all the genes evolve at the same rate? Here's a plot of the evolution of 3 different proteins, cytochrome C, hemoglobin, and fibrinopeptides, with the millions of years since divergence on the x-axis and the number of changes. And you can see that the rate of evolution for these guys is different. It's also true that in the pro-insulin molecule, the C-peptide, which is lost during maturation of insulin, evolves at a different rate from the A and B peptides that are tied together with disulfide bonds in the mature protein. And so you can have different rates of evolution in different domains within a protein. And those, again, we make simplifying assumptions that don't always hold. Another complication that I think is really important for looking and thinking about divergent proteins. So let's say we have an ancestral gene, and we see two variants of that gene in the contemporary sequences. And we see that there are 3 sites at which there are differences from the ancestral gene. Well, that sounds well and good, but if in the pathway of evolution from here to here, we end up with more mutations accumulated, some of them through a second nucleotide, and some of them actually going back to the original position. We don't know what happens here, and that's what we try. We try to compute all those possibilities when we build a tree. And that's complicating, and it ends up that we can end up with distances between genes where there's a matrix, a scoring matrix called the PAM 250 matrix, which corresponds to pairwise percent identity between 2 proteins of around 25%. It is similar to the Blossom 62 matrix that's used in BLAST, and it infers that there are 250 nucleotide changes per 100 residues. So it takes into account all of these changes, but we don't know which ones they were. So again, behind the details, when you push the button and you get the tree out, all of this other stuff is happening. And I just wanted to bring that to your attention as we move forward, because I'm going to say a lot of things today that are kind of simplifications of the reality, and it's important to realize that bioinformatics, as we're talking about it, is a very inexact science. And it's a little bit problematic because we have a tendency to have very nice websites that are extremely intuitive to work with, not all of them, but a lot of them. And we put our data in, and we push the button, and we get an answer out, and we assume it's the right answer. But there are all these complications that can happen while we're getting there, one of the reasons that we end up with high levels of misannotation. So, back to the main point, sequence analysis provides access to genomics-related information. We work typically with the sequences themselves instead of keywords or names because there's lots of problems inherent in that. And you can do searches for exact matches or nearly exact matches. I'm not going to talk about any of this today. I'm really going to talk about near and distant homologs in multiple species. So this is kind of the outline of what I'm presenting. And I'm going to tell you a little bit about sources of sequence and structure data, how we identify homologs, and especially divergent homologs, using sequence and structure. Once you get a set of homologs that you think are pertinent to the question you want to ask, you really have to cluster that data in some way, whether you make a multiple alignment, or you build a phylogenetic tree, or a protein similarity network, and I'm going to talk a little bit about all of those. And this is the real proof of the pudding here. What you want to do is map the conserved elements of sequence and structure to conserved elements of function. And that's where it gets hard. That's where it takes expertise. That's where even framing the questions in a big era data actually, I think, is a very challenging problem. I have, near the end of my talk, a listing of some orthogonal approaches, other kinds of information that inform functional inference. I may not get to those in my talk. I just gave you some primary methods and the original and a few other references to them. They're very important today because homology is not enough anymore. We really have to use many different kinds of information. And there's a lot of the computational community that is devoted to trying to develop more sophisticated algorithms for weighing that information. And you may have seen in databases you use that you get a confidence value on the answer. Often that confidence value refers to how well the algorithm worked, not how good the answer is. And so I don't know how to tell you what confidence to put for a particular biological problem, but we're really trying to figure out how best to bring together all this kind of information. And lastly, something that is sometimes left off of talks by bioinformaticists, but my training is originally as an experimental enzymologist. And ultimately, we have to do an experiment to tell whether we're in the right ballpark even if we can only do one experiment to tell us something about a fairly large set of unknowns. Excuse me. So I'm not going to spend a lot of time on these. These are just a listing of some sources of sequence data and annotation. When Gemma gives her first workshop later today, she goes into these in a lot more detail. For those of you who can't be at one of the workshops, I'm at least listing some of these. There are a lot of different kinds of databases, including thematic ones that are important depending on what kinds of proteins you're studying. For classification of protein sequence information, the way to go now, unless you have a very specialized problem, is that there are very large sets of pre-computed solutions for many basic bioinformatics questions and problems. And one of those is a site like Interpro, which is supported by the European Bioinformatics Institute. Gemma will talk more about that today. They try to classify sequence space into families of homologous proteins. There are many kinds of predictions that are made there, and there are a lot of member databases and resources that are used in those predictions. And that's all I'm going to say about that now, but you really it's really worthwhile to look at this because, for example, PFAM has a set of, has compiled all the known sequences into families, provide probabilistic models that you can use to test whether your own sequence, where it might fit. You can search there. And when we start with a big superfamily of 48,000 or 80,000 sequences that we're going to work on, we download the set from PFAM, cross it with the rest of the information from Interpro, and then go from there. And that means we may decide some of those proteins are really, we don't want some of them. Maybe we need a second model from PFAM. But we'd spend all of our time just gathering the sequences if we didn't do that. I also wanted to mention a really important aspect of classification in bioinformatics that you may be familiar with, but you may not have thought about why it's so important. And that's the gene ontology. So the gene ontology comes from a community collaboration that tries to give consistent descriptions of gene products and their relationships to each other in different databases. And it has three kinds of functional information: molecular function, biological process, and cellular component. And many of the databases, for example, in Interpro, our small structure function linkage database, many others, use evidence codes based on the gene ontology so that people can identify what kind of evidence is available for certain functional assertions. So here are some of the evidence codes that the gene ontology has. You could, for example, say, I want to know all the enzymes that have experimental assay data, and just look at those. Or at least you may want to use those as anchors when you're making inferences about other sequences that are related, but that have been annotated using computation alone. So it's really important to have that kind of information. It also means that people who are developing resources can get this information in computer-readable form so they can handle very large amounts of data and put it in the right places in the database so that it's actually usable in a high-confidence manner. So, identification of divergent homologs. I'm going to talk about sequence-based methods and structure-based methods both. Structure used to be kind of a special afterthought, but we now have so much 3D structural information and it is such a valuable resource. I think for the last several years, it's been time to incorporate three-dimensional structure information into our basic bioinformatics workflows. And I'm going to show you some examples of that as we go on. I'm going to give you just a little bit of the underlying theory for how we do the simplest of these operations, which is pairwise alignment, just so you have a feel for how this is actually done and what it means. Some of you may have seen this before, but I think it's instructive. So how do you find a relationship between a query sequence and one or more additional sequences? Well, you can align your query along each of those 50 million sequences in the database and ask what's the best alignment between those, and is there a statistically significant answer, and if so, it's reported. That's what you do when you do a blast, for example. Then formalizing that problem is what you do here, and the requirements for getting the report is you have to have a way to evaluate the matches and mismatches between any two characters when you do those alignments. You need a score for insertions and deletions because one protein may have an insert relative to the other, and so you have to try to find an alignment that works well with that. You have to optimize the total score, so you add up the score for every one of those presumably inferred independent positions, which we know aren't independent, but that's the way we do it on a large scale. And then you have to have a way to evaluate the significance of the alignment. Scoring systems, represent the degree of match between two letters. Simplest is identity. And by the way, it's typically not a good idea to report sequence similarity in a paper because unless you define what your model for similarity is. So it could be, I'm just going to use hydrophobics versus hydrophilics. I'm going to use different kinds of chemical properties. There are hundreds of these scoring systems to define similarity. We typically report identity because that is defined. And similarity is something you can think about as a next step, but as a primary representation, we report identity. So we have definitions of similarity that are based on some kind of chemical or biological physical properties, and those include the Blossom matrices, which is what are the default matrices you use when you run a blast, say, at NCBI or many other places. Score is a very simple statistic. You ask, what is the probability of the residue I see in a particular place in my alignment relative to what I would see, of seeing that relative to what I'd see by chance? And that's a setup, that's the basic and most fundamental statistic used in sequence analysis. So there are many types of scores that are typically derived from evolutionary models, many other ways that are to do this, and I'll tell you a little bit more about the more sophisticated ones in a minute. Just in case you've never seen it, this is the Blossom 62 matrix. And what you see is a sequence, two sequences aligned, which is what you do in BLAST, and you give a score for the match between the sequence in this column and the sequence in this column. So if you have a cysteine, and you see a cysteine in that same position in your alignment, it gets a score of 9. An alanine with a cysteine is 0. Alanine with alanine is 4. So the positive score varies depending on how likely it is to see those kinds of mutations. A tryptophan, which is seen conserved very long range, typically in transmembrane domains, has a positive score of 11 if you have a tryptophan in the same place. That's a big signal. And when you're doing conservation analyses, you realize that you're not just looking at what's mutated or changed in column 1 versus column 2. You're actually looking at properties that are typically seen, the likelihood of what's seen given some empirical analysis. But if you change a tryptophan to many other things, like a proline, you get a pretty bad negative score. And so that's actually how the Blossom 62 matrix works, you add up all those scores, and then you say, what's the best score I can get? And that's done using matrix algebra, and then you report that score and evaluate its statistical significance. For database searching, the only criteria you have to judge how good the likelihood of an evolutionary match is an estimate of statistical significance, because you have a huge amount of output and you need some way to start sorting through that data. Now, typically, some of the most interesting problems are down in the noise. They're not very significant. And so you want higher resolution methods to try to tell whether those matches are good, and structure is one way to give you that. BLAST, just for those of you, I'm pretty sure almost everybody here has probably used BLAST, but just to give you a tiny bit of information, it's one of those major heuristic algorithms that speed up searching. It's not optimal, but it's pretty close to optimal, and it's actually very good at this point, especially some of its variants, like SciBlast. The current version actually incorporates gaps. That wasn't the case in the original version. It was just local alignments only. And it does all of the things I've been talking about. I'm not going to spend time going through these. These are sort of some of the main variants of BLAST. There are actually lots of others. This one is actually really nice because you take a protein sequence, T-BLAST-N, sorry, against a nucleotide sequence database. It translates all 6 reading frames. It's a good way to pick up mistakes in actually calling a gene. So you'll see ORFs that are too short to be real, and it's a nice way to pick that up because you don't want to do that on the scale of thousands and thousands of sequences, but it's a useful, there are a lot of useful kinds of things you can do here. Extending our reach, what we really, beyond the sort of basic BLAST, which uses these lookup tables to score, we typically are using probabilistic models instead. SciBlast is one version of that. Instead of using a scoring matrix like Blossom, you actually use a specific, position-specific matrix that is tailored to your data. So you do a BLAST run, you find the best scoring homologs. From that, the program looks at all of those sequences as if it were a multiple alignment, and then says, at position 10, what are all the changes I see? And what is, given those changes, what is the probability that I'll see one of those? And so it's a weighted scoring matrix that's tailored to the data itself. It's a very kind of simple hand-waving way to explain that. It's really explained best mathematically, but I'm not going to go into that today. You can build these PSSMs, they're called, directly from SciBlast data. SciBlast is actually very good for finding remote relationships. It has its problems that I'm not going to go into today, mostly if you're trying to find very divergent hits. Hidden Markov models, which I'm sure most of you have heard of, is a different way to build these kinds of probabilistic models, again described only appropriately using the mathematics. These are used very, very broadly for classification of new sequences. The PFAM uses hidden Markov models to define its families. We use in our database hidden Markov models to define superfamilies, subgroups, and families. You can come in with a sequence, search against our HMMs, and we'll tell you which of the groups your sequence best matches to, as long as we had that group in our database. There are also variations and software available. This is actually a combination of the logo kinds of approaches, but using hidden Markov models. So what you see here is by the size of the letters at each position, the likelihood, essentially a summary of the probability of finding that letter at whatever position you are in the sequence alignment.
That's a quick question. So,
yeah.
Like for BLAST versus SciBlast, are there specific reasons to use SciBlast
Yeah, if you're trying to find more divergent proteins, it works well to use SciBlast, and it has been used a lot for doing exactly that. The problem is that usually when if you're trying to gather a great big set, it may help you find all the proteins in your set. You may want to do something like download the PFAM set, which is just domain only, and then use SciBlast, starting with, say, a query and adding new sequences because you want the ones that came in yesterday or something like that. The problem with SciBlast is that you start with a single query, but it then generalizes that query to the whole set of sequences that are first found. Those profiles can actually drift depending on what's in the database. And so there are issues if you're looking for really distant relationships. I think there are better ways to do it, but they're a more complicated experiment. I think if you have a new protein, I would definitely use SciBlast because it will give you, and you're looking for more distant relatives, it'll give you a lot of those. You still have to evaluate whether you believe it. Did that answer your question well enough? These probabilistic models, as I said, are really the way this is done these days, and there are lots of pre-computed solutions at the Uniprot database and others. I've listed 3 of the main databases that I think are really important that use probabilistic models and hidden Markov models, state-of-the-art methods to try to gather sets of sequences that are, sometimes they use also structural information. Motif analysis, I'm not going to spend much time on this, but sometimes you just want to find, maybe the data is so diverse, you just want to find the particular motif that is important. Now that's a tricky situation because this is where you can get into finding convergently evolved proteins or non-homologs. So we did a study a few years ago where we analyzed the entire thioredoxin fold set of superfamilies. So many of those proteins have a cis-XX cysteine motif that is important for the redox chemistry because they all have a thioredoxin fold. It turns out that from our very large-scale analysis of all those superfamilies, which included thioredoxins, glutathione transferases, peroxyredoxins, glutaredoxins, all these different superfamilies, only about 60% of them actually have that cis-XX cis motif. The rest have some degenerate version or harder to recognize version. So motifs are a two-edged sword. In the radical SAM superfamily, you can find that motif because it's really required for the binding of SAM, but it can come from different folds. And in our study of this, and Gemma could tell you more about this later in the methods, we're having trouble figuring out quite how many folds there are because these proteins are so distant, we can't really compare them very well. There are very few structures. And so this is a little bit of a two-edged sword, but it's also very useful if you're looking for, and I'll show you in a minute an example, maybe you can't get a good multiple alignment because the sequences are too diverse to really align them well, but you can identify some important motifs. So I put this in to the talk because there are a number of useful algorithms for doing this, and I mentioned some of the different kinds. We really like MEME, which is a statistically based method that, given a set of sequences, will identify motifs and give you some kind of statistical significance value for them. They're trickier to score because they're typically short regions. So from sort of the information theoretic content of database searches, you need a certain number of bits of information, that is, a certain amount of information to be able to evaluate the statistical significance of a match between one protein and lots of other proteins. When you have only very short segments, and this can apply to de novo sequencing by mass spectrometry, for example, sometimes those segments are pretty short. And so how to evaluate whether you've really found a good match what happens is that there's more variance across the e-values or the p-values that you get. And so it's a little bit trickier to evaluate because they're just short. There's not very much information in them compared to a full-length sequence. I want to talk a little bit about protein structure comparison. The requirements are very similar to those for sequence comparisons. You need the same kinds of scoring. You can do structure versus structure comparison, including motif type comparison. I call fine structure here. That might be comparison between active site motifs in 3 dimensions. And you can do sequence structure comparison as well. So, sorry this is faded out. I should make a version of this with a white background, but it's a very old slide and I haven't had time to remake it. The bottom line is we can see further with structure than we can with sequence. And what I mean by that is that at a lower level of similarity, you can see some structural similarities where you cannot pick up sequence similarities. And that's a critically important thing to be able to do if you're looking for very divergent homologs. Here are 2 proteins that if you can't see them from the back are highly superimposable that to the naked eye look like they're very similar to each other. There's a phosphonattase that cleaves the phosphorus carbon bond, and there's a haloacid dehalogenase, 2 very difficult chemistry reactions for biological systems to do. These 2 sequences of these 2 proteins are less than 15% identical. So you're way down in the noise level. And if you I ran this a couple years ago, but I think the results will be broadly similar. If I run it against Swiss prot, there's 2 sequences to try by blasting 2 sequences together or using one of these as a query and running a blast, I find the right hit that's the 106th hit, and that's way down in the noise. It's way down in the noise. It's actually not in the noise level. It has a pretty good e-value, but it also hits some non-real homologs before it hits that one. So it's hard to sort out what the right answer is. If you run this against GenBank, you get 0 hits. To the right, you don't find this if you use this as a query, is the bottom line. But they're very readily apparent that they're homologs from, or the inference is that they're homologs from the structure comparison. So this is sort of the slide that is comparable to the sequence similarity slide. I'm not going to spend time on it because I want to talk about a lot of other things. But I want to make the point that when we do structure-based alignment, we don't have access to evolutionary theory. And that's because there is no evolutionary model for the transition between one position, a position of a residue in space relative to the position of another residue in space if it's a different residue. So we don't have a model for mutations, basically. And the other problem that happens is that 2 structures that may be very similar to each other overall may have loops that are quite different in length. And so it's tricky how to set the gaps, which is also tricky in sequence, but it's even more tricky in structure. And the other problem is that 2 parts of a structure that play a particular role, for example, bringing a substrate in the right orientation in one structure, may have evolved to have that region of the structure play a rather different role. Maybe it's a protein-protein interaction. And so those are very hard to evaluate, those kinds of alignments. Still, ignoring structure, I think, is much to our detriment because there's such a rich amount of information there. So I've just listed here some of the methods for fold-based comparisons, and that's what most of our structure comparisons are. They're comparing the alpha carbons of a fold in 3D space and asking how well do those match. You almost can't use these algorithms for some structures where there's lots of varied inserts, because it's just really difficult to sort out what the core is. If the two proteins have pretty similar cores, you could compare the folds fairly well. But that doesn't tell you anything. If you're talking about an enzyme, that doesn't tell you anything about the active site. And the active sites can have moved around, not hugely, but often in ways that are subtle, that have the important information about differences in mechanism. So again, it's tricky to do this. One way to solve that is to align two structures around particular residues, like active sites. And there's a nice tool in UCSF Chimera that will let you do that. And so you might actually get the folds where they're kind of twisted relative to each other, but you really care about how those active sites align. And I'm telling you about this because, again, you can't just use any of these programs just out of the box. You have to say, how am I framing the question? What do I really want to know? Is this bioinformatic approach going to tell me what I need to know? And I think that's a really important lesson. Bioinformatics experiments are experiments, just like assays are. And you have to run controls, and you have to think about, are you using the right tool for the question you have to ask? And that seems really obvious, but it's pretty easy to push that button and get a really nice answer out that says, here's, oh, you have very high confidence result. And you just tend to believe it, and don't do that. Okay. All of these are really quite good algorithms. If you try the same alignment with each of them, you get a slightly to fairly big difference in the answer. And that's a really good way to know that it's a tricky situation. These two methods actually are pairwise alignment methods that are very fast. And I'd say they're pretty good, but they're pretty approximate too. But we use them for our structure-based networks when we build similarity networks, because we're doing pairwise comparisons sometimes of 150 structures. And we can't wait 2 weeks to do that. We need to do it much faster than that. And so it, again, depends on the problem that you're trying to deal with. This is a really nice paper that evaluates structure-based comparison. And if you're going to do this, I would, this is a really good review, and it'll give you some ideas about what are the best methods and how to evaluate them. So we also have, in addition to databases like PFAM, which classify the sequence universe according to protein domains, we have classifications of the structural universe. The one I have been the most familiar with for many years is SCOP, Structural Classification of Proteins, and that has been used principally in the United States for a long, long time. There's been quite a lot of manual classification that went into this by a guy by the name of Alexei Mirzin, that I remember one of the reports of the first CASP contests where people were trying to predict structures. One of the people got up at a report and said, well, we're all depressed because we all use these great methods and Alexei got everything right and we missed most of them. Well, it's not quite true anymore. That methodology has gotten much better, but that's where the manual classification actually comes from, which is Alexei Mirzin. These classify structures according to three-dimensional comparisons, and then there are sequence databases that are associated. These kinds of classification resources serve as primary gold standards for comparisons and algorithm developments. Since Gemma Holliday came to my lab, her work was done in the UK, and in particular, her postdoctoral work was done in the European Bioinformatics Institute. They've used CATH, which is a different classification system that works enough different from SCOP that it's a little bit hard to get your head around the two. This is a really excellent resource, and I've come over kind of to the dark side because Gemma's really convinced me that I ought to be using this. SCOP also has not been officially updated for 5 years, although there is a new version of it. And if you go to this link, it'll take you to the new prototype SCOP. They lost their funding for a while. It's been very difficult to fund some of these bioinformatics resources, even the ones that are used by hundreds of thousands of people a year, just because funding hasn't kept up with the need for these tools and these resources, because nobody could go out and do this stuff. You know, even a lab like mine that does this full time, we can't spend our time trying to gather a set of 80,000 sequences and verify that they really belong together. We go to PFAM, download theirs, and then evaluate that. So there is now a new data, I can't remember the name of it, there's a new position at NIH devoted to these kinds of resources. And I think that's going to change things quite a bit, because I think the experimental community really needs this kind of information. So I've got a couple of other things listed here that you can read about if you look at the slides. So I know you can't see this in the back. There's two nice structures here that look very similar to each other. And so you might ask, why use sequence information at all when structure really works so much better? And I think there are quite a few good reasons for that. One of them is the proportion, the number of structures is very, very much smaller than the number of sequences. The ratio is around 100 to 1 or worse, and it's just going to keep getting worse. You could see it's diverging. And even though we're much better technically at getting a lot of structures, we're never going to get close. We can also use homology models, and they have become really very good, down to even about 25 or 30% identity, depending on what algorithms you use. It's so good that, for example, Matt Jacobson has been able to build homology models of proteins, do in silico docking of databases, metabolites from the KEGG database and other metabolites to those models, and make the right functional prediction that can be validated experimentally. That's really, really good. And if you look at his homology model relative to the structure that was then characterized afterwards, it's really spot on. It's very, very close. So the methodologies have come a long, long way. But even with that, we're still going to have a huge amount of sequence information that we don't have structures for. And especially as we get into unusual habitats and start pulling in real outliers in metagenomic data, it's going to be hard to get structures of these guys. Especially since maybe you can get the sequence, so you can then synthesize the gene and try to express it, but that gets deep fast, and a lot of those guys live in really weird environments that it's hard to know what their culture requirements are. And trying to express them in normal organisms is tricky. The other reason to use sequence is, for example, I was talking about motifs. So this was an early study of these proteins in this haloalkanoic acid dehalogenase superfamily, which now has about 80,000 sequences. Originally, we could not get a good alignment of a non-redundant set of those sequences. They were just too diverse. What we could do is identify 3 motifs that were highly conserved and that we could map to structure superpositions. And you see that those residues are nicely overlaid in the 3D superposition of those structures. So here you at least, even though you don't have a really good alignment and you don't have a really good tree, you could say, well, these seem to be a few of the highly conserved regions, and it makes sense they look like they're part of an active site, and we actually have chemistry that tells us the importance of this nucleophile. And so put together, you could really use the sequence and structure together to give you much higher quality information than you would get if you just use structure. The other reason, besides having so much more sequence, to fill in. So you might have two structures, but they might be so far apart that you don't know what other kinds of variations happen sort of between the properties you see in one structure and the other, and sequence can sometimes fill that in. And the other reason, of course, is we can't build trees that make sense in terms of evolutionary models with structural information. We just don't have the scoring matrices to define what mutations we expect to see. So clustering. That's the next step after you find your homologs. And I spent a long time on finding homologs, and then I said, just go to one of these databases and download them. It's often a little more complicated than that, but it's a pretty good answer. But hopefully you have a little bit of a feel for what it takes to actually find these homologs, and even if you run something as simple as BLAST, what you're actually doing and how those are scored and some of the strengths and weaknesses of those approaches. So there are lots of ways to do clustering. And what I mean by clustering is you take your set of homologs, and now you want to ask, which guys look more like each other? Are there classes of sequences that are different from each other as groups? Does that tell us something different? How do we map that to information we already have? So I think you may already know this, but I think it's a point that is often forgotten. And this is really the best way to evaluate sequence comparisons that you get out of straightforward programs like BLAST. So you can use these alignments, and I'm including alignments generated by hidden Markov models, which are the highest quality in terms of the methodology that's used. You could do screening for membership in a family or a superfamily. You've got a new sequence, you want to know where it fits. If you run it against a database of families or superfamilies, it might tell you which family it belongs best to, and that will save you weeks and weeks and weeks of work. Identification of conserved elements, important to function. I'll show you really the huge difference a multiple alignment can give you relative to a pairwise comparison. A number of other important aspects, speciation over a multiple alignment space, where you kind of have two sequences that may be very different from each other, but if you've got them in a multiple alignment that kind of connects those differences, you can kind of see where some of the changes are that might distinguish different groups that you care about. They may have different functions or different mechanisms. There's a bunch of issues and caveats with these, especially for divergent sequences. It's very hard to produce a single multiple alignment from correctly aligned subsets of inputs. That is, there's usually more than one good solution, and it's hard to tell, sometimes without structure information, which is best. There's a sensitivity to the number of sequences used. I think, what is Clustal Omega is 2,000, Gemma? You could use 2,000 sequences in Clustal Omega. Now, you could take a set of sequences that are 50,000, and you can filter them into bins where you can say, in each bin, I'm going to put all the sequences that are 50% or more identical. And then you get the number of comparisons of bins down. You could take one representative from each one. That can be fraught with all kinds of issues depending on variations of the sequences in the bin. If you have to go down below 40% to bin them, then even your pairwise alignments are starting to really degrade badly. And so there's all kinds of reasons that that the number of sequences used, there are methods to build a 1,000,000 sequence alignment, but I can't think of, you can look at it with a computer, but a human being can't do that because it's all gaps. So anyway, and there's sensitivity to the specific sequences. I'm going to give you a couple of practical examples. And this is a very simple one, but I think it's an important observation. So this was a BLAST that I ran a number of years ago. It doesn't matter when I did it. I took that phosphonatase sequence and I ran it against the database, against SwissProt, and it gave me a hypothetical protein at a not quite statistically significant value, or close to statistically significant, but not a high quality value. And the first protein that actually was known was a phosphoglycolate phosphatase. And it didn't have a great, this is actually seen in p-value. It's not a great p-value. This is the alignment showing highlighted the identical positions. So the alignment between this query and this known, the blast alignment is shown here, 21% identical. So that's not very good. The fact that the alignment is global through the whole sequence gives you some confidence that this may be a real match, even though the significance is not great. But even at 21%, how do you tell what are the most important residues? How do you tell which ones are important for function? Now, if you have an alignment that's 60% identical, you can have a lot more identities, and then you really don't have an idea. So that's what you get from a pairwise alignment.
So, Patrick, when you said match here, are you speaking of how they do have the same function?
No, I'm just saying between each position in
I
the...
understand that, but you said, like, how do you know that they match? I mean, how do you define match? Like, are you trying to find a protein that catalyzes?
Well, I actually wanted to know what... Originally, we didn't know how... This was work, again, with Debbie Mariano. We didn't know how this worked. And so we were looking for... This is a number of years ago. We were looking for proteins that were similar, that we understood, so we'd get a better idea of how it worked. And so the first thing we did was run a blast, because there weren't very many tools at the time besides this. And that's what this data... This is actually from that experiment. So the closest match, that is the sequence that matched at the best score that had a known function. Sorry, I should have spelled that out. Thanks for asking. Was this phosphoglycolate phosphatase. And that's what I'm showing aligned here. So, as I said, how do you tell what's important? So here's our little motif alignment that I showed you earlier. And here's the phosphonatate sequence, and here's the phosphoglycolate phosphatase sequence, which is the same as this. And here now, in the multiple alignment, it was very easy by eye to figure out what residues were the most important. If I map those back onto my pairwise comparison, there's no reason I would pick these out if I looked at it in the blast data. And this even isn't identical. And it's not identical because there is a difference in the requirement of the, there's a shift base that's required in one of these, and there's a difference in the mechanism that it works okay because of the nature of the substrate. You never know that from this kind of data. Now, you'd never know any of this if we hadn't been working with Debbie, who knew something about the mechanism of these proteins, and that's where we get back to experience and experiment, which is you've got to have some of that deep knowledge. But this is kind of how the interplay between that multiple alignment and a pairwise comparison works. And so that's a really important thing to notice. And you want to do this kind of clustering, whether you're doing a multiple alignment or a phylogenetic tree, or you're downloading a set of pre-computed family proteins that are essentially give you something that looks like a multiple alignment. That's what you want to be able to look at. The other thing that at the time, we only had one structure in this superfamily, and it was the haloacid dehalogenase, and that's shown in the context of this multiple alignment of the motifs, and that's how we were able to map these active site residues. And knowing that structure and knowing something about the mechanism told us how the phosphonates worked by comparing it to the rest of the proteins here with all the other knowledge. And that's really putting the sequence, structure, and functional information together to solve a tricky functional inference problem. And again, this is how a few years later, they were able to map to structure. A couple of other issues. One of them is weighting. Most of the alignment programs handle this for you themselves. The Clustell family does. You want to have, if you have 20 sequences you're aligning and 15 of them are more than 90% identical and all the others are quite different, you could get a big skew in how they're aligned unless you weight those sequences to remove that kind of redundance in the algorithm. It doesn't happen so much anymore because the modern algorithms do a lot of this for you. I'm not going to spend any time on this, but I'm giving it to you because it gives you the basic papers and some of the main kinds of multiple alignment methods. The one that we're using now is Clustell Omega. So Clustell W does not work very well for divergent proteins, and that's one of the very common ones that's used. This is a new representation of Clustell that we like very much better. Some of the other really good methods that muscle was really the state of the art for a number of years. I think Clustell Omega now beats it out, as does methods like MAFT. So this is a group of methods that you might, if you're interested, you may want to look at some of those references. Gemma will be talking about some of this in the first workshop. Structure-guided multiple alignments. So this is a pretty specialized area, and there are algorithms and software that incorporate some structural information. At the end of the day, if you have a group of proteins that you're really trying, that are very diverse and that you're really trying to sort out how the structures are put together to vary function, you have to kind of do this by hand. You have to do the structure-based superpositions, and then you have to use that information to guide your multiple sequence alignment. And this is a project with Dan Hirschlag on the alkaline phosphatase superfamily. And what I'm showing here is there's a core domain in all of the proteins in this superfamily. And then these domains are decorated by a variety of complex inserts, some of which are not even related to each other, so we don't think they're homologous. And they can occur in somewhat different positions in the core domain. And so that's a very complicated problem. But we wanted to know, we really wanted to tease out in as accurate a way as we could the differences between the proteins that use one metal, like the sulfatases, and the proteins that use 2 or 3 metals. That was important to our overall study, and it was important to the sort of general mechanistic issues that Dan was working with. So we aligned these core structures, identified where the inserts were, and a lot of this was a combination of algorithms that were out there and a lot of handwork. That was actually a big part of a graduate student's thesis. And then came up with this structure-guided multiple alignment where we can identify all the metal binding ligands, the nucleophile, and some of the other important ligands that differentiate these sulfatase-like proteins from all of the others. And this was a really major aspect of guiding this work. 3D motifs, there are also ways to build motifs in 3D space. They give you, we call it fine structure. They give you nice views of sometimes functionally important residues. There are a number of algorithms available. I'm going to tell you a little bit about 2 of them that actually use both sequence and structure information. One of them is active site profiling. This is work from Jeff Skolnick and Jackie Fetro. It's been around for a long time, but they have just developed a new version of this that will be published soon. What they do, and I think this is a really nice way to think about the problem as well as a good approach for a tool, is they identify in 3D space residues that are important to function. And those may come from a structural study where they're actually in the literature. There are ways to do this automatically, but they tend to be not quite as good. This comes from experimental data, which is really nice. They then project each of the positions of the alpha carbons for these residues that are involved in function in 3D space through to all of the other residues that are within a certain number of angstroms. And they pull out those little motifs. As you show here, here's the one for the blue residue, here's the one for the green one, etc. And they make they catenate all those little motifs, and they make a catenated string, which they then use to search databases and compare these proteins. And it actually, we've used some of our gold standard superfamily data from our database to evaluate this method, and it does almost as well as manual curation that took us years to do in the case of some superfamilies. So we're very excited about that approach and that collaboration. So another one is called evolutionary trace. This is work that was initiated by Olivier Lictarge. He's done a lot of improvement and extension of these approaches since he first published this. Basically, he builds, I'm sorry about this twirling. It was made by a graduate student of mine at least 7 or 8 years ago, and I cannot get it to stop. I just have to make a new version, but sorry about it. I forgot that it does. I forget, I don't want to think about it, and then it turns up on my slides. So he builds a dendrogram, which is not a careful tree. It's just a pretty reasonable tree, and then he identifies and sets aside all the positions that are conserved in all these sequences, and then looks for patterns that differentiate subsets of the alignment. And this is a nice way to explain how multiple alignments work. And then he maps each of these positions that are colored differently on the structure, so you can start to see how they overlap and what distinguishes these different subgroupings, and they may represent functionally different families. This works actually very well for sort of medium divergent proteins. Again, when you start to get to really distantly related proteins, you just have to put in a lot more work, is the bottom line. The real proof of the pudding, ultimately, is mapping the conserved elements of sequence and structure to conserved elements of function. And that's actually the primary goal of my lab, to do that. And one of the problems here is we have really good ways of defining similarities here and similarities here and mapping them to each other. When we get to function, we tend to wave our hands and say, well, here's what the function is, but what's the quantitative comparison of a reaction or a reaction step? And that's work that Gemma is heading up in my lab to do that kind of analysis. We've been really good at comparing ligands and small molecules for a long time, because this was developed by the drug development industry. And so that part of the equation is there. But I am going to tell you, just to set the stage a little bit for the very last part of this, is how this applies to functionally diverse enzyme superfamilies, which my group has worked on for a long time. So this is sort of follows a model we call chemistry-constrained evolution, where structural elements involved in mediating some fundamental aspect of chemistry is conserved across divergent proteins, and then the ability to bind new substrates and do different overall reactions evolves. And the poster child for this comes from work a number of years ago, before I even joined the project, which involved John Gurlt and George Kenyon, Greg Petsko, John Kosridge, Dagmar Ringer. You may remember this work from a long time ago, where they discovered that mandolate racemase and mucinate lactonase enzyme, which are two very different reactions. They have very different enzyme completion numbers. Here are the structures superimposed. They're very, very similar, as are the active sites. And that sort of eventually led to this model of chemistry-constrained evolution. And I'm going to talk about these kinds of functionally diverse superfamilies as an illustration of protein similarity networks in the last few minutes. For our database, we then use that as a way to hierarchically classify enzyme superfamily data sequences at different levels of granularity to which we can map different levels of detail of functional properties. So we could define a superfamily as containing a subset of active site residues, and these are conserved across all 27,000 members of this particular superfamily. This is the enolase superfamily, actually. These are the residues that are conserved that all of those proteins have. They're associated with a particular partial reaction and stabilization of an intermediate, and we can identify the signal in sequences in the 27,000 sequences we have. So any sequence we find with this pattern, we can assign to that superfamily. So that constrains the search space a lot for functional inference, because now we know something about the basic chemistry and even some substructures in the substrates. We then use sort of traditional bioinformatics methods and resources to break all those sequences into smaller groups just so we can work with them. And it's only when we know the residues that distinguish one reaction family from another do we assign sequences with that reaction family. So we don't annotate at this level of granularity unless we have good supporting evidence. And that's how we try to avoid misannotation. I think this kind of gives you an idea of how we use this kind of information for this kind of a problem. This is also the sort of primary principle underlying how our database is set up. So, what do you do for clustering large-scale data? Functionally diverse enzyme superfamilies represent more than a third of the universe of enzyme superfamilies, so it's a lot. And they can be all kinds of different fold classes, as shown on some of the superfamilies we've worked on here. We can estimate how many reactions might be represented by looking at the reactions we know, looking at the unknown space, seeing how different that unknown space of sequences that we don't know the reactions, how different they are from the knowns. And then there are 27,000 sequences here. There are 80,000 sequences here. CoA hydratase or crotonase superfamily, nearly 50,000 sequences. Isoprene synthase, 16,000 sequences, more than 50,000 products. These are big sets. How do you get your head around these? In each of these cases, we have good evidence that there's a conserved active site architecture linked to some fundamental chemical capability, whether it's a partial reaction or a capability such as use of an oxyanion hole, for example. 10 years or so ago, we were tackling this for the aminohydrolase superfamily when we only had 5,000 sequences. We have, I didn't put how many are on here, sorry. We have many, probably tenfold as many, at least 50,000 sequences. And what my postdoc did was she went through and she clustered everything at 40% identity and binned all the sequences that were 40% or more identical to some other sequence in a bin. And then she made all of these different cluster trees for 17 major subgroups where it was color-coded by known function and gray were unknowns. And we looked at it and threw up our hands and said, we can't deal with 5,000 sequences. Look at how complicated this is. We're going to work with every one of these, and we don't know how they relate to each other. So this was really the motivation for starting our work on protein similarity networks, which was trying to cluster this large-scale data. So what we do here is, this is a network that shows you the similarities. Each circle here, each gray or colored dot is a sequence, and the lines between them represent some level of similarity. And we could color all the positions, all the sequences where we know something. We could color, highlight a sequence that maybe is an unknown we want to know about. And this is now becoming a sort of major tool in the arsenal. There are at least 7 new servers to make these kinds of networks that have been published in the last couple of years. So what are these networks? If you look at a tree, you could see how, if you had 3 functional classes, you could see how a tree might be like, say, an evolutionary trace. might do a pretty good job of separating these into 3 groups. You can also use graphical network models to do this, where every circle we call a node is a sequence or a structure or a ligand or something else. And we can set a threshold to draw an edge, where we could say, I have to have a p-value or a blast e-value as good as some statistically significant threshold cutoff to draw an edge. And if I have a very permissive threshold, like e equals 1 times 10 to the minus 2, I have a hairball. And that doesn't tell me very much, because if I don't know what all the colors are, I can't tell which function's which. As I make that threshold more stringent, they start to separate. And if I'm lucky, I'll find some threshold at which I've actually separated functional classes. Now, this doesn't happen very often, because it turns out that functional boundaries don't track well with sequence similarity or structure similarity boundaries when you start to get to divergent proteins. And there are lots of reasons for this that I don't have a lot of time to talk about, but this is a very difficult problem. And so having a way that we could build many of these networks for thousands and thousands of proteins, map them with all kinds of functional information, is a very powerful way to get started in building new hypotheses. I'm going to show you very briefly an example with the glutathione transferases, where we started with greater than 13,000 non-redundant sequences. The number of total sequences is much larger, but this represents the more than 95% identical space. As you all may know, glutathione transferases do lots of different kinds of chemistry. They're very important in redox biology. Here's the sequence similarity network for the whole glutathione transferase superfamily, thresholded at a blast e-value of 1 times 10 to the minus 13. So what we did is we filtered all of the sequences into bins of 50% identity or better. The median alignment length is 210 residues. Well, these tend to be about a little over 200 residues long, so we're getting good global alignments. The node colors are defined by the classes of glutathione transferases, alpha, beta, mu, sigma, that have been used by that community for 70 years. They don't represent specific functions. They represent groups that have been worked on, that there was some inference that they might have some similarities. Sometimes that they, like all these guys with the reddish colors, all came from eukaryotic systems originally, although now we know there are some bacterial sequences in those groups. The thing that blew us away, and this has just been published in PLOS Biology, so you can read more about it if you're interested, is how little we know. Almost all of this is gray. There are 30,000 references to GSTs in PubMed. Huge amount of effort gone into this superfamily. This typifies every single superfamily we've looked at. The huge amount we don't know. If we require a more stringent threshold to look at that same network data, here is an e-value of 1 times 10 to the minus 4. So we require more significance to draw an edge, and that means a lot of the clusters fall apart, and we have a bunch of singletons that don't have aren't connected to any other sequence if you require that much significance in the score. And we mapped it again to these Swiss prot classes. You see that there are big clusters that no one's ever done an experiment on. The number of specific reactions that have been characterized as far as we could tell in the literature, and that was using Swiss prot as a proxy on a large scale, and as much literature as we could look at. This was a collaboration with Richard Armstrong and Steve Almo. There are like a few hundred out of these many thousands of proteins. There are only a few hundred that we actually know what they do specifically. So we built this set to match the classes as best we could that already existed, to try to suggest what new classes, sequence classes, might look like, so people could start thinking about what targets they might want to look at. Especially, you could say, well, I'm going to take this network and I'm going to map all pathogens to it and see which GSTs are in pathogens. And that might be a handle I would want to start looking, characterizing new GSTs that no one had looked at before. So that's the kind of thing we do with this. We also build structure similarity networks. This was about 130 structures. What's nice about this is it told us kind of how some of these interior relationships work that we don't see in sequence because they're just too far apart to connect with sequence data. And so that's another adjunct that's important. There's a lot of pros and cons of these, lots of advantages. They're fast. We see multiple connections that we don't see when we build trees. We could, even a very simple metric like BLAST captures strong conservation signals, and we have validated the BLAST data. If you use lots and lots of queries that are very diverse, we get correlations in the distances that are pretty comparable to what we get for phylogenetic trees and hidden Markov models, which take much longer to compute, of course, and we could do that as well. Lots of caveats. Networks are not trees. They're tricky to interpret, and we're working on a paper to try to tease out some of those important ways that we think about it and sort of let people know what kinds of issues there are, both strengths and weaknesses. I'm not going to have time to talk about orthogonal approaches. There are many, and as I mentioned, here are some of them that I have a few slides on that add other kinds of biological information. You could then go back and map that kind of information to this sort of data. And we get a lot of this information in an automated way from other databases, which is one of the importances of having these large resources, because otherwise we just can't manage the data. We couldn't possibly look at all the GST literature ourselves. Not that the automated methods are necessarily as good. And then I want to end with this point, and that is, despite the fact that we've got an awful lot of data and exploiting it is really, really valuable, we can't afford to be disconnected from experiment. I'm part of a big group that is trying to work toward development of methods and algorithms for automated function prediction. And I keep saying every one of the superfamilies we've looked at is different in terms of how it's set up to deliver function. And there's no number that says above 60% you can transfer annotation. I could give you lots of exceptions to any of those rules. Trying to figure out how much experiment we need and how well covered we need to make these superfamilies, when you see how much unknown space there is, I actually think is a very important question for the kind of inquiries we want to make today. And hopefully you can see how a lot of the pieces I've tried to bring together for you, albeit a very small amount of the material, how that fits into this kind of analysis. And I want to leave you with this. I know this is an overwhelmingly experimental lab, and it's certainly my roots, and I think we still have to come back to this. So I think that's where I'll end. Thank you. I know you all want to get to lunch soon.
We have time for a couple of questions.
Randy, looking at these divergent sequences, specifically at the identical residues, are certain amino acid residues, they have certain weights when you're looking at the identities? And the reason why I ask this is because I found that glycines and proteins tend to be really well conserved across divergent species due to the structural features that they have to offer.
Yeah, you're absolutely right. And that's why Blossom Matrix has more weight on those residues. For a positive match, I know it went by really fast, but a positive match gets a higher score. And the way you ultimately define the whole score for the alignment is you add up all those scores. And you know, some of them are negative values, so you subtract that. There's a lot of methods out there. I'd say most of them are approximate. There are many kinds and patterns for protein interactions. Protein DNA interactions are somewhat better specified in some ways and far less specified in others because your alphabet in the DNA is much smaller. And so you get a lot of redundance, so that's going to be embedded in the secondary structure that you can't infer necessarily from the sequence. But yeah, there are a lot of methods. I'm happy to talk to anybody else, and we're going to try to go over a lot of this in a series of workshops. So, thanks.
Okay. Any announcements? Yeah.